Welcome, everyone. It's a pleasure to continue the invited lectures of the Josep Carreras Research Institute against leukemia. And today we have the pleasure to having with us Dr. Maria Casanova-Acebes. She's a junior group leader at the Spanish National Cancer Research Center, CNIO, in Madrid, where she leads the Lab of Cancer Immunity since early last year, January 2021. Maria studied biology at Universidad Autónoma in Madrid under the supervision of Dr. Andrés Hidalgo of the CNIC. Her thesis was about neutrophil aging and clearance, where she made seminal contributions. And from there, she moved as a postdoc to the lab of Dr. Miriam Merat at the Mount Sinai School of Medicine in New York, under the support of IMA Frontier Science. During her stay there, she did great discoveries in the ontogeny of macrophages in different tumor types, from lung, breast, ovarian tumors. And recently, also, she has studied embryonic macrophages and tumor outcome. This was last year. And her contributions to the macrophage ontology and myeloid dysfunction are the reasons that she's here for to explain about also her past research, current and future research, probably. And she has been invited already to many lectures in Keystones, in the European Congress of Immunology, received several awards, ACR AstraZeneca award, etc., and the Ramon y Cajal, Chris Cancer Research. So she's an emerging young scientist in Spain, and she's going to explain today about the role of myeloid cells in metastases using different technologies and mouse models. Maria, the floor is yours.
Well, Manel, thank you very much for this super nice introduction, and thank you for having me today. It's a pity I cannot be there physically, but hopefully maybe next year I can show you even more data on the projects we are.
Maria, just to remind everyone that there is a question and answers button here, that you have questions, you can use it. And Maria, please go ahead.
Okay. So what I'm going to tell you is a little wrap up of the last findings I gathered while I was still a postdoc in Miriam's lab, and the three main research lines that we are covering in the lab. And of course, one of my favorite cells are macrophages, and I know that they might be not so popular in the field of immuno-oncology, but I got very much fascinated by them because I guess the fact that they can modulate so plastically their transcriptome really catch my attention. And these cells were very early discovered by Elite Mannikov, and using a very rudimentary microscope, he observed that these cells are very highly phagocytic, and depending on what they eat, for example, apoptotic neutrophils, as we found while I was doing a PhD, they can trigger either an immunosuppressive or an immunomodulatory phenotype, for example, when they eat debris from the cancer cells, as you can see in this confocal image. And while I was a student in Andrés' lab, I rotated in Miriam's lab. And at that time, it was when the boom of plasticity, particularly macrophages, was starting to be discovered. And as you know, macrophages rely on different cytokines depending on the microenvironment they live. And for example, lung macrophages rely on GM-CSF and PPR gamma signaling, whereas if you take macrophages, for example, from the peritoneal cavity, these macrophages will rely not on MCSF, not on GM-CSF, but will rely on retinoic acid and another transcription factors such as RXR and GATA6 to be called a macrophages. If you remove any of these survival factors or any of these transcription factors, those cells forget that they are a macrophage, and so on and so forth with every single macrophage you find in your body. And there's another major difference in macrophages that makes them functionally different, and this is their origin. Either macrophages arise from the embryonic compartments when we are developing a baby in our bellies, and these macrophages have the uniqueness of being self-maintained, or macrophages which arise from the bone marrow, which are very much short-lived and then return, and the turnover of these cells mostly is on a daily basis. So again, These are only some of the hallmarks of macrophages plasticity, but I borrowed this kind of graph from cancer biologists talking about these hallmarks of heterogeneity, particularly for these cells. And this is just to summarize you here how macrophages behave differently either in homeostasis or inflammation, because the factors that they receive for this imprinting that I was mentioning to you before is completely different in this situation, and because also the interactions that these cells establish in these different two scenarios are quite different. And emerging hallmarks that we are starting to appreciate in this heterogeneity are the ontogeny, the energetics these macrophages utilize, also their response to, for example, treatment, particularly to immune checkpoint blockade, and also the state, the evolution of these macrophages, as I will show you now, or the evolution of these macrophages, either if you are looking at a primary tumor or a metastasis. Not only that, those macrophages live in a human body, right, that maybe is experiencing other preconditioning diseases such as aging, obesity, recurrent viral infection or bacterial infections, viral infections such as in the recent case of COVID, which are going to imprint also some epigenetic and transcriptomic changes in macrophages. So those are things that also we have to take into account when we think about how we modify these cells. And also this is one of the reasons by which this plasticity has challenged cancer researchers over the last 4 years. I'm going to just go over this over this slide very quickly, and again highlighting the complexity, diversity of macrophages, this plasticity. Now we take it in mostly in cellular states, but we have to still really understand how long those states are lasting in particularly diseases, how they contribute to disease progression or suppression, and how do we target these cells actually with a very much rational behind it while we maintain their homeostatic functions. And again, what are the long-term consequences, for example, of depleting some of these populations? So something that I'm sure you already know is that macrophages have been involved in growth, immunosuppression, and just genesis, invasion, and metastasis. But the tumor stage in which these macrophages appear and how these macrophages change over time, or if novel population of macrophages appear at these stages, have not been looked so much into attention. Importantly, in the case of human, most of the studies have been based in vitro. Now, with the advent of single cells, now we are single cell technologies, we are moving into the in vivo studies. But I still find fascinating to still read many manuscripts in which they really only highlight this plasticity and they summarize it to the M1 and 2 paradigm, which already we know that this is not recapitulated in vivo. So one of the main results also of presenting today this work here is that this definition is incomplete because the residency, the lineage from which this macrophage arises is also so much not taken into account. Okay, so this is what I'm going to tell you today. So, and this is the way we start this project. So we arise the hypothesis that macrophages of different origins might modulate tumor immunity. And we decided to focus on non-small cell lung cancer because the lung allows us the opportunity to look at the same time a compartment which arises from embryonic progenitors of macrophages, which are alveolar macrophages, which self-maintain, and at the same time macrophages which again are constantly recruited into the lung, in particular into the tumor, by circulating monocytes, which locally differentiates into macrophages. So we wanted to know what is the different contribution of those two cells, because we think that we can help to better identify what is the tumor we are in front of, and we can actually better treat these tumors if we know what is the ratio of these so different populations. Okay. So the models we utilize preclinical models, utilizing a cell line, that is a model of non-small cell lung cancer, KP, which is KRAS mutated and p53 deleted, and that we have also engineered to express GFP so that we can follow the tumors and the growth of these tumors by imaging and longitudinally. And we also analyze using single-cell RNA-seq human samples of non-small cell lung cancer patients of stage 1 and stage 2 with the similar mutations. So just to show you again and coming back again to the big punch I made on the heterogeneity. We didn't find N1 and N2 macrophages. What was very fascinating to see is that we find an enormous heterogeneity of macrophages that we basically classified in 4 different clusters. But when we analyze, and this is a heat map of the major expressed genes that identify these different clusters of cells, this I did it together with a fantastic computational student in Mira's lab, Andrew Leader. What we identified, again, were not these arginine genes, these TF-beta genes, as the canonical ones that we identify normally in these M1 and 2 studies. And again, way more diversity than it was previously anticipated. We also wanted to understand if this kind of diversity was somehow identified in mice models so that we can utilize those as avatars of different functional questions. And that's exactly what we did. This here, what you are seeing is a summary of the heat map of the genes that are conserved between human and mouse, and we could then later explore functionally. So for example, in this cluster 1, we observe that the MRC1 or CD206, normally classified as an M2 marker, was identified both in human and mouse, Siglec1, Marco, PPR gamma, and so on and so forth for the different clusters of cells that we identified, different clusters of macrophages and monocytes in the process of differentiation towards the macrophage lineage. But still, this didn't tell us nothing about the origin of the cells. So for that, what we did is the CYAN experiment in collaboration with Boris Reisis at NYU, in which we took advantage of a tool he developed a couple of years ago that allow us to track cells that are only uniquely of hematopoietic origin. So for example, by the induction of the recombination of the MAP17 gene using tamoxifen and following up the cells that recombine express TD tomato, over the lineage, we observed that cells that derive, we could detect cells that arise uniquely from adult HSCs. Normally, this gene is never expressed or recombined in cells which seed the tissue before, meaning that cells which are tissue-resident macrophages do not become red, so to speak. So we control this. We observe that normally cells that we know that derive from hematopoiesis are mostly recombined, such as neutrophils, inflammatory monocytes. And what we notice in the lung is that a population that we name tissue-resident macrophages become barely populated by cells that recombine with the TD tomato protein. And this is consistent with the work of Simon Jonah and Martin Guillian that studied tissue-resident macrophages in the lung in the steady state. So then we move into the cancer setting and we repeat the experiment. Normally we had to wait quite a lot because these stem cells, right, take a lot of time in order to develop a full differentiated compartment. So we had to wait 6 months till getting like a nice and efficient labeling of the cells. And we perform single cell RNA-seq, separating between the cells which are tomato negative, meaning cells which are from embryonic origin, and tomato positive cells. And to our surprise, we observed that in tumor-bearing mice, still there is a compartment of cells which are from embryonic origin that show barely contribution of TD tomato cells, while the most majority of the other cells are from bone marrow origin. Then, retrospectively, we compared the frequencies of these clusters with the analysis that I showed you before. And what we observed is that in both in mice and in human, there was a dramatic reduction of this compartment of tissue-resident macrophages, and that allows the expansion, indeed, of the bone marrow-derived compartment. So next, we wanted to understand what is functionally this population of cells doing at different stages of the development of the tumor. And for that, we look for markers that allow us to unequivocally identify these cells. So it turns out that the expression of Siglec1 in combination of MRC1 allow us to detect this population that you see here overlay with a reporter mouse. We generated CD169 TD tomato together with CD206. So these cells that you see here, orange, those are the tissue-resident macrophages that are located mostly outside of the tumor foci. So these two markers, and again, in combination, we cannot use only one marker to identify macrophages, allow us to, in the lung, to identify this population of MACs in KP tumors. So then we follow these tumors longitudinally, and what we observe is that indeed, when we analyze the tumors very early, when the cells are seeding the lung, we observe that this population of cells are in close contact to these macrophages. So these are the cancer cells in green, and these alveolar macrophages with kind of the shape of a fried egg. Those are the alveolar MACs. And as long as these tumors progress, what we observe is that these macrophages like somehow form a structure, kind of a granuloma towards the tumor foci. This occurs also in the genetic model developed by Tyler Jacks, the KP model, and we also observe that there is a reconfiguration of these alveolar macrophages, and these are the quantifications of how these resident macrophages are located either in the intratumoral or peritumoral foci. So next, we wanted to understand how this occurs molecularly, and for that, we reasoned to analyze these macrophages when they are starting to form these kind of structures. So what we did here is bulk RNA-seq in order to really understand which is the molecular program we observe in these lesions, and it turns out that these macrophages, both at the RNA level, but also epigenetically, conform a set of genes that are much more involved in the remodeling activity with a set of metalloproteinases and peptide antigen binding. And I will come back to this long term later on. So then, in order to functionally mimic that remodeling scenario, what we did is taking these tissue-resident macrophages, isolated them from tumors, and compare with this remodel capacity that they had with MOMACs, with the other clusters that arise from the bone marrow origin. And what we observe is that following them by imaging and following these spheroids they form, is that these macrophages form invade way more GFP area. So somehow reminiscent of their activity of remodeling while they are forming an alveoli during embryo development. When we have had a closer look into these spheroids, what we notice is that indeed what they do in these cancer cells is deplete these cells from ikaderin, as you can see here in the quantification, and this does not occur with another population of macrophages derived from the tumors. These spheroids also generate way more invasive protrusion, so somehow suggesting that in vitro, at least these macrophages promote kind of an EMT model. EMT process. This is just a nice video I always like to see because these macrophages are highly motile, and this is again reminiscent of how they move towards in the alveoli, scanning for bacteria and virus, but they also scan for other pathogens such as cancer cells. And as you can see here, some of these macrophages take up some cancer cells, and you follow these cancer cells longitudinally, you will see that these cancer cells generate a novel organ. So then we move into an in vivo setting, and for that we took advantage of CD169 DTR model, in which the instillation intratracheally of diphtheria toxin allows the depletion of cells which express CD169 gene. So that what we observe is that when we can quantify using confocal imaging, what we notice is that in these cancer cells at very early stages, we observe a downregulation of canonical transcription factors involved in EMT, such as TWIST and CEB. This is a quantification that you can see here. And it turns out that as a result of this early broad invasive phenotype, what we notice is in day 30 tumors is that the tumors are way more reduced. As I was mentioning to you before, we observed that these macrophages, they have a division of labor. They are not also involved in the remodeling of the lung parenchyma while a tumor is sitting, but we also noticed that the class 2 molecules were much more upregulated. So in order to understand that, we had a closer look into the T-cell microenvironment that we observed in these tumors, and we observed that there was a dramatic reduction of regulatory T-cells in these tumors, as you can see here, and quantified by imaging. And that is conversely correlated with a massive infiltration of T cells. And here I'm quantifying total CD3 T cells, but most of the T cells that we quantify here, and actually in our manuscript, are CD4 T cells. And that also correlates with a better anti-tumor immunity response. Interestingly, when we get rid of these macrophages later on, so once that the tumors have formed and once that this capsule is somehow impeding the accessibility of T cells into the tumor microenvironment, we observe that the depletion of macrophages did nothing at all. Not in the case of T cells, not in the case of promoting and reverting this anti-tumor immunity. So just to summarize these findings and move into new data, what I show you today is just there's like kind of a choreography between macrophages in the tumor microenvironment, particularly in KP tumors. We notice that these macrophages at very early stages are the ones that dominate the lesions and are involved with this initiation, remodeling, and immune suppression. Some dormancy maintenance is something that I know the lab of Julia Aguirre is starting to explore, and we are collaborating with them. But later on, once that the tumors somehow have been encapsulated, in this case, this the hematopoietic compartment and the inflammatory cells which take over completely. So there are different waves of these ontogeny distinct macrophages which have never been looked before, and these tissue-resident macrophages are mostly relevant during the tumor inception. So the clinical relevance of these findings is the early integration and maybe activation of this compartment can really be used to promote a tolerogenic potential to reduce this tumor progression at very early stages. Okay, so now I'm going to move into different topics that we are exploring. Some of them, of course, emanate from these findings that I couldn't completely finalize, and some others come back also to some studies I did while I was a PhD. So I showed you before that when we get rid of this population of macrophages, we see a massive infiltration of mostly CD4 T cells. So how this activity can modulate T cell infiltration is something that we didn't cover at that time. And something that was also very, very interesting to see, and that I was working with another talented PhD student, that Britta in the States, is that as long as these macrophages encapsulate this tumor, between this capsule of cancer cells and macrophages, there is a capsule that is like a line of fibroblasts, right? And that indeed is what shields the tumor for this T cell infiltration. So interestingly, what we see, and I started to do these experiments while I was still in the States, where I had a lot of mice and I had a lot of resources to ask many questions. I noticed that when I get rid of the same population, and now what I'm looking is at the stroma, not immune cells, I noticed that gating on cells which are CD45 negative, epithelial negative, and endothelial negative, the rest of the cells, so the stroma of the cells, these are pericytes, there are also some vessels, but this population, which express both podoplanin and PDGFR, is enriched in fibroblasts. So the lack of these tissue-resident macrophages in this model promotes a significant reduction of this population of fibroblasts. And not only that, what we notice also is like some inflammatory markers that are used by the laboratory, for example, of San Ontarley to define the activity of these cells are also massively downregulated. And this is not a matter of all kinds of macrophages, because when we use models that have an intact compartment of embryonic macrophages, but are lacking more macrophage, monocyte-derived macrophages, what we notice is that the stroma compartment remains the same. So it's, again, something specific that is triggered by the resident compartment. So then the question was kind of obvious. What does it happen when you challenge this lung with a tumor, in order to understand how does this stroma react? So what we are starting to see is that indeed there is another population of fibroblasts which appear and that is actively proliferating. This population is expanded, this other not. We are now trying to identify if this population using fate mapping models for fibroblasts actually downregulates these markers. So this population gives rise to this one. But interestingly, what we see is like kind of the opposite of what we are seeing in the steady state, that these molecules are somehow way more inflammatory. And as you might remember, what we see in this model is that these changes promote better infiltrations of CD4 T cell responses. So we are generating now using CRISPR different fibroblast models in order to knock down these genes and trying to address that using spheroids. Another very interesting project that we have in the lab, and that indeed is part of an ERC project I wrote last year, is trying to understand how circadian rhythms are connected or disconnected from the host rhythms in the tumor microenvironment. And this is something that really caught my attention from some experiments I did back in the past with Andres, in which I observed like BMAL total deficient knockout mice have a way more exacerbated tumor burden. And some other experiments I was doing while I was still in Andres' lab, we were very much obsessed about time in that lab, is that when I transfer these 16 cells, a melanoma tumor that I inject in the tail, those go directly into the lung at different times of the day. This is what it means. CT is a German term that means Zeigeber time. So Zeigeber time refers to any kind of thing that you give a stimuli to mice in this case after certain hours. For example, CT4 corresponds to 11 a.m. CT8 corresponds to the 3 p.m. and so on and so forth, covering the 24 hours of the day. So just to speak briefly, what we notice is that the burden of these tumor cells, which was much more exacerbated in the morning as opposed as in the afternoon, suggesting that the rhythms of the host somehow determine changes. And indeed, what we wanted to study this is that not only the rhythms of the host determine changes in the tumor, in the defense against the host, but also cell intrinsically, the rhythms of the cancer cells are also promoted changes. And this is what we want to ask in this project: how is the time sense in tumors, and can we take advantage of these vulnerable timings for therapy? And very interestingly, this is data that we had fresh from the oven with a computational student in the lab, Gonzalo Soria. This is the transcriptome profile that we have analyzed using Circadian, a tool we developed in the lab of Andres. And this is analyzing only the cancer cells that have been seeded into a mouse at different times of the day, as you can see here. And you can see that there are some programs which are way more expressed in the morning. This corresponds to CT5, so noon, as opposed to, for example, at night, CT13, CT17, or CT21. And this is like, somehow contrary to what is thought in the field. Normally, the cancer cells are known to lose the circadian regulation, but in this case, in the KP model, what we have noticed is that indeed we have actively and very nice fluctuating BMAL1, which is the key transcription factor, which controls circadian timing in every single cell of our body. Doesn't matter if it's a cancer cell or if it's a cell of your retina. So nicely fluctuates, and this is in opposition to the levels of PER2, which is the repressor of this BMAL clock. So we are very nice now interested to understand which are these programs, and indeed, as a disclaimer, I can tell you that what we have analyzed so far, and again, this is really data from 1 week ago, so please take into account that we are still refining this analysis, is that the cyclic genes that we observe in the morning are mostly related to glycolysis, hypoxia, and mTOR signaling. So again, suggesting that the metabolism of the cancer cells might be way more exacerbated in the morning when the immune system of the mice is way more inactive as opposed as at night. So we are starting to think some ways to target this pathway. Finally, and this is, I think, the last slides I'm going to show you, I'm very much interested as well in neutrophils, and what we identify is a molecule, TRM1. You might be aware of the signaling of TRM2 in macrophages, but what we noticed in a screen that I also developed in the lab of Miriam while I was still a postdoc there, is that there is a molecule, TRM1, which becomes way more expressed in metastases, particularly on neutrophils. And this molecule agonizes with DLR2 signaling and DLR4 signaling, and some antibodies are in phase 1 clinical trials, particularly for metastatic solid tumors. So we have noticed that these cells express mainly this molecule in neutrophils. There is also particularly in the lung some expression in alveolar macrophages. And what we have observed is that when we transfer these different models of metastasis, such as the KP1 in total deficient TRM1 knockout, there's also a massive reduction in the tumor burden. There is a better anti-tumor immunity. And we are now testing different metastatic models, for example, in collaboration with Lara Alvaro and Manuel Valiente in the Brain Metastatic Lab at CNIO. We are using the models in order to see how these metastases in the brain grow. And these metastases are very much populated by neutrophils, and not only by neutrophils which express TRM1. The same thing occurs in collaboration with Kathleen Rosalén and Tony Celia. Sorry for this because it's very blurry, but when we analyze metastases, breast metastases that seed the liver, we observe that the vast majority of those neutrophils upregulate TRM1. So now we are trying to identify how this TRM1 becomes upregulated most preferentially within it might be by soluble factors that arise from the primary tumor. And if this TRM signature, which is associated also to IL-1 beta and arginase signaling and some prostaglandin signaling, particularly in the lung, is something that occurs specifically in the tissues, or it involves also the reprogramming of the bone marrow when they see that the tumor is arising. So the main questions we want to answer here is can we reprogram this TRM1 associated signature, and whether this program is really tissue imprinted, or is this reprogramming arising from the bone marrow? So with that, I think I made you a pretty vast coverage of myeloid cells. So I just want to acknowledge the people of my lab, our collaborators, the animal facilities at CNIO, and of course, the funding that allowed us to do all this research. Thank you very much, and I'm looking forward for your questions.
Thank you very much, Maria, for this presentation, to introducing us macrophages. I know they're popular or not. I think they're pretty important. It's a different thing. You know. But again, thank you very much for the presentation. So I remind all the attendees that they can use they can ask questions using the questions and answers button there. You have it in place in your screen. So I compile a few of the questions. And here there are some examples that I would like to translate. So one relates to the circadian. Sorry. So do you know that there are some data that suggests that the time of immunotherapy given to the patients is relevant to the response? So this makes sense with your data? Or?
Yeah, I mean, we have to take into account that humans are diurnal species and mice are nocturnal, right? It was a very nice study published in New England Journal of Medicine. It was called Memoir, and they give melanoma advanced patients, stage 4, if I remember well, in the afternoon after 4 p.m. and before 4 p.m. And the ones that were given 4 p.m. ones respond way better than the ones that would respond that were given anti-PD-L1 in the morning. So again, suggesting that this actually is not like ciencia observacional, something's like, you know, some curious thing. It's way different. It's not. When I was doing the PhD, I was still like somehow rejecting this idea, because I say, okay, it's just number of cells. But now that we can really see that the phenotypes, and we have done also some quantification, I don't want to bother with so many flow data, but at least in mice, we notice that, you know, the effector T cells and some NK cells are way more active in mice at night, I suppose, as in the morning. So it is something that matters, the state of the immune.
Yeah. I think it does. It does. So just a curiosity, just a comment that years ago we identified that some of these circadian genes were silenced by DNA methylation in cancer. So some tumors that are losing BML1 or period 2, they don't have the expression, so they do not follow circadian anymore. The tumor itself.
Yeah. Did you identify it like it was all in vivo, or was it in vitro?
In patients.
Okay. I mean, we are of course, like, you know, I want to make sure that this is not something related to this cell line, and of course we are focusing on lung cancer or the different ones just to make sure that is relevant. I didn't also wanted to show you more data, but when we knock it down, now we are knocking down Bimal, and we got that sequencing again, but we haven't analyzed it. So we partially lost this glycolysis thing. So we are like somehow excited of that, but of course we have to validate that if that's the case in certain tumors in human, of course.
And here is a question by Maria Fidalgo. It says, is there a way, a specific marker of distinguish the bone-derived macrophages from resident macrophages in the bone?
In the bone, I mean, in the bone is kind of difficult because in the bone, the vast majority of macrophages are indeed bone marrow derived. And in this case, CD169 corresponds to the bone marrow derived. So you can, the vast majority there, so they are not resident macrophages. So using CD169, you will identify bone marrow derived. In the case of the lung, CD169 identifies the resident ones. So that was, my disclaimer was, be careful, you have to look into the tissues on a tissue, you have to look into the markers on a tissue-specific manner as well. The best combination we found, and the always experiment you have to do, is fate mapping. It's annoying, it's painful, but it's the experiment you are going to be asked to completely say, okay, this is the origin of the cells.
Another question, in this case by Elisabetta Mereu. Say, hi Maria, nice talk. In your data, have you identified the interstitial-like monocytes macrophages?
Yeah, I mean, all those, like in the 4 clusters I show you, this cluster 2 is you can still break it out in 3 different clusters, and they're the interstitial macrophages, and 2 of the ones that are the GPMB kind of macrophage and the TREM2 macrophages. So the interstitial macrophage, so you call it interstitial, but you have to locate them in the interstitium. But the TREM2 and the GPNMB do not necessarily reside in the interstitium, although they are bone marrow derived. It's a matter also of now all the spatial transcriptomics in the lung, which is a very, very rich myeloid-rich environment. It requires also some location studies to be done together with the transcriptomics to the single cell.
Yeah, I think we'll take some answers from the experiments for sure.
Mm-hmm.
Here, a question is, what about the field of targeting macrophages themselves in cancer therapies?
I mean, you know that many of them, the field has targeted macrophages using anti-CSF1, anti-CSF1 receptor, anti-CCR2, to impede the recruitment. And of course, most of them, particularly in glioblastoma, have shown resistance again. So the trend now is really looking into the molecular in their reprogramming, right? There are some PY3 kinase inhibitors that have been used in order to, let's say, reprogram to diminish the suppressive potential of macrophages. You cannot get rid of the cell because again, then if you get rid of alveolar macrophages, for example, you will die of a viral or bacterial infection, right? But you can use of their signaling. There are very nice work also from Anne, which is Carmen Andreas Snitzer, which they are using this kind of train immunity, so applying low dose of agonists, LPS agonists, in order to reprogram, and this is because epigenetically you change that macrophage, but you cannot make them give them an stimuli so abrasive that will cause actually the depletion of this resistant compartment and the MOMAC replacement, which are way more inflammatory, right? So we have to be careful to train these cells in a way they maintain their homeostatic functions, or maybe just it's only one subset that is the one we have to target. But we are still trying to identify in alveolar macrophages. There are 5 clusters, and for example, there is one particularly that shows a lipid metabolism very much exacerbated with interferon signaling very much exacerbated, and we are trying to tell apart that cluster, but it's very tricky because we don't find markers to isolate them. That's also annoying.
Yeah. And the one I have here also is it relates to the calves the cancer, the fibroblast. Because it is known that in lung cancer immunotherapy, you have a lot of fibrosis. These are more resistant tumors to immunotherapy. So can you do a little bit, again, in this relation between the CAFs and the macrophages?
Yeah. I mean, something we are trying, as I was mentioning to you, the ones we identify more are like close to these fibroblasts are also very rich in lipid metabolism. So we are starting, instead of depleting these cells, we are trying to give like some inhibitors of this lipid metabolism in order to see changes in fibroblasts. But we have just started these experiments very, very recently. So we don't have the mice are running basically. So that's the only thing I can tell you so far. So, but it's interesting because, these macrophages, of course, they are a source of TGF-beta. So we thought about TGF-beta at the very beginning, but we are aware of as well of how TGF-beta signaling, how spread it is in the tumor microenvironment, the failures they have caused also when they have tried to combine it with immunotherapy. So I think, again, it's a matter of targeting the proper cell and maybe more specific signaling pathways. If you go to TGF-beta, all the healing system, you are going to be also very much in trouble. You're going to, maybe impede the tumor, but the healing of the lung, which requires this signaling, is going to be destroyed, basically.
Okay, so I will take the last question in a more personal way here. So in this, you say that this is very extreme, no? Talking about M1 or M2, no? It's something a simplification of, and you have many intermediate states there. So, of course, I can think that these are epigenetic states there. People have looked in detail there. This can be modified with drugs. What are your thoughts about this?
Yeah. I mean, there was a couple of years ago, in which they were doing HDAC inhibitors of transferases together with PY3 kinase inhibitors in macrophages, in alveolar macrophages. For example, their interferon signaling was dramatically changed. So the thing is that, okay, how can you deliver those drugs specifically into the macrophages? And, it might be other macrophages that they have this signaling. So something that we were also thinking is like, when we think about the nasal vaccines, that is much more in favor. I think we have to go that way. So we want, these are the cells which are somehow perfect in the way that you will touch those cells and you won't touch other macrophages. So that's the kind of thinking we are approaching. So some of these resident compartments, they are always like somehow in accessible areas. They are really they are the guardians of mucosa, and sometimes in most cases in the gut as well. So I think that would be the way. So a delivery that local takes advantage of the location of the cells.
Okay, thank you very much, Maria. Interesting time. We should close it here. Again, very thankful to the speaker. Let's give her a clap. It's an online one. Okay, and hopefully to see you in a presential manner soon here. Thank you very much.
Thank you very much, Manuel. Thank you very much, all of you.